to finish, we are now going to talk about how to transport large molecules and using exocytosis and endocytosis. In the beginning of the class, I showed you examples of this. Small molecules crossing the membrane. And now what we're going to study is what happens when you have big molecules, yeah? When you have polysaccharides, really large molecules. So you will require those transfer will require a process like exocytosis, pinocytosis, phagocytosis, different type of processes that in all these cases require energy. So first thing that I wanted you to remember is that in the previous sections, we talk about how small molecules like CO2, oxygen, and water enter to the cell. And we also talk about some proteins, some carriers, some pumps. However, now we're going to move to large molecules like polysaccharides and proteins, and they do it via vesicles. So they use vesicles and that they require. So this is the third thing that you have to remember, that they require ATP. They require energy.
Here we have a vesicle in this diagram, in this schematic, and you see that the vesicle is migrating to the membrane and then it fused to the membrane and then release these particles, blue particles. And here is the same thing that you see in this diagram. You see, but instead of blue particles, you see green particles moving to the membrane and this releasing this protein. And this process is called exocytosis. As I said, the vesicles migrate to the membrane, so first migrate, then fuse, and then release their contents outside of the cell. And this is exocytosis. There are many cells that use exocytosis to export their products. Endocytosis, on the other hand, is the opposite process. In this case, you see that there is a lot of molecules outside of the cell. The membrane is going to have this little pocket that is going to start growing and then is going to go inside of the cells. So in endocytosis, the cells take macromolecules by forming vesicles from the plasma membrane, and endocytosis is the reversal process of exocytosis and involve different proteins. And there are three types of endocytosis. First one, phagocytosis, then pinocytosis, and receptor-mediated endocytosis. And this is a very important process. Why? Because, for example, if we want to, in the future, cure many diseases, we need to put new genes. So for example, we have new DNA here. And one way to introduce that DNA inside of our nucleus is maybe to use endocytosis. So take this piece of DNA that goes into a process of endocytosis, and then it can be released inside of the cell. There is still a lot of work to be done, but we start learning in more detail how the cells can get molecules inside of their nucleus, and endocytosis is an important process that we will study.
In endocytosis, membranes invaginate or pinch in to form a vesicle, moving the enclosed materials inside the cell. This process can take different forms, each involving its own specific cell machinery. In phagocytosis, or cell eating, the cell engulfs debris, bacteria, or other sizable objects. Phagocytosis occurs in specialized cells called phagocytes, which include macrophages, neutrophils, and other white blood cells. Invagination produces a vacuole, which usually fuses with one or more lysosomes containing hydrolytic enzymes. Materials in the vacuole are broken down by these enzymes and degraded. In pinocytosis, or cell drinking, the cell engulfs extracellular fluid, including molecules such as sugars and proteins. These materials enter the cell inside a vesicle. Epithelial cells in capillaries use pinocytosis to engulf the liquid portion of blood at the capillary surface. The resulting vesicles travel across the capillary cells and release their contents to surrounding tissues, while blood cells remain in the blood. Unlike phagocytosis and pinocytosis, receptor-mediated endocytosis is very specific. It is triggered when membrane receptors bind to specific external molecules, such as protein cholesterol complexes or proteins bound to iron. Membrane vesicles pinch off, and the external protein and its cargo are brought into the cell.
For any questions that you have. Have a good day. Bye.